We are going to conclude our study of cells and genetics today with Unit 8, Lesson 8, and we will be learning more about Punnett squares. Now, here's our warm-up. Capital A, lowercase a. Would that be homozygous or heterozygous? I'll pause for you to answer. If you chose heterozygous, you are correct. Homozygous would be the same, so you'd have to have 2 uppercase or 2 lowercase. Heterozygous is 1 uppercase and 1 lowercase. Our goal today, you should be able to define heredity as the passage of genetic instructions from one generation to the next, and be able to predict the possible genetic outcomes for offspring by creating and using Punnett squares. And our overall question, why are Punnett squares used in the study of genetics? Both of our standards are supporting standards. And our only vocabulary today is Punnett squares. You will need a piece of paper for this lesson because you're going to be drawing out Punnett squares and filling them in. All right, so Punnett squares are used for predicting possible genetic outcomes for offspring. They help to calculate the probability that certain genes will be passed on or not passed on to offspring. Doesn't matter if mom or dad's genes are on the top or the side, because you put mom up top, dad on the side, or vice versa. And capital letters are always written first if they belong in the box. So if you have a capital letter, you always put it first. We're going to start out with an introductory video.
Welcome to Moo Moo Math and Science. In this video, I'd like to look at Punnett square basics, solving some Punnett squares. Before we start with some Punnett squares, let's review some basic genetic vocabulary. A trait is a characteristic passed from one generation to another. For example, eye color. An allele is a different form of the same trait.
We say allele.
For example, eye color is the trait, and the allele would be the different colors of an eye. A homozygous dominant genotype is written with 2 capital letters because a dominant trait is always written with a capital letter. A homozygous recessive genotype is written using 2 lowercase letters because a recessive is written with lowercase. A heterozygous genotype is written using 1 capital letter and 1 lowercase. Here are the steps for setting up a Punnett square. Draw the Punnett square, write the genotypes of the parents on the side and on top, and then fill in the boxes. Let's work a couple example problems. Let's go ahead and work some example problems. This first problem says, let's say that in cells, the gene for the length of the whiskers has 2 alleles. The dominant allele, capital W, codes long whiskers, and the recessive allele, smaller case w, codes for short whiskers. What is the probability of producing offspring that have short whiskers from a cross of 2 long whisker cells? One that is homozygous dominant and one that is heterozygous. Okay, so let's go ahead and follow the steps. And obviously we have already drawn the box. And now let's go ahead and set up the parents. Well, one parent is homozygous dominant, and it says to use the W. And homozygous meaning they are the same, so I will have a capital letter because it is dominant, and I will need to write 2 of these. So homozygous dominant is capital, capital. And the heterozygous will be a capital letter and a lowercase letter because they're different. Okay, so we've done step 1 and step 2. Now we just fill in the boxes. And let me show you a little bit how we fill in the boxes. And so that you can see that this is a lowercase letter, I'm just going to put a mark through it. So this capital letter goes here. and this one goes down to here. And on this first one, I'm going to go really slow. This next one, it goes, follow the marker, goes here, and this one goes there. And now we repeat on the bottom. We have a lowercase w, and then this w moves down here. And now this moves all the way across, and this one moves down. Okay, so now we have the box filled in. And so we have, next what we do is, the only way you will get the recessive trait is if you have two lowercase letters. So in this, you're going to get long whiskers, long whiskers, long whisker, and a long whisker because both of them have a capital letter. So,
Okay, can you spot what he did wrong? He did not put the capital letters first here. You always put the capital letter first.
the chances of getting long whiskers are 4 out of 4, which is 100%. And then this would be 0 out of 4, or 0%. Okay, that's our first example problem. Let's work one more, and this one has something that's a little tricky, but we can figure it out. It says if one parent seal is heterozygous, long whisker, and the other is short whiskered, what is the probability that the offspring will have short whiskers? In this one, let's use the letter B, okay, and lowercase b. So first, let's write out the parents. Well, heterozygous, that's pretty easy to write. We're going to have capital letter lowercase. But now here's the question. Which one is dominant, the long or the short? Well, it told us that before, but if you didn't know that, anytime you have a heterozygous genotype, it always is the dominant trait. So we know that long whisker is dominant. And then short whiskered is going to be, the only way you're going to get short whiskers is if you write homozygous recessive. And now we fill in the boxes. Okay, this goes here. This B goes here. This B goes over here. This B goes there. This one goes down. And finally, this one goes all the way across, and this goes down. So now, in this case, we're going to get the dominant trait, because remember, anytime you have a capital letter, you get the dominant trait. So we have 1, 2 dominants, so that's 2 out of 4, or 50%. And then the recessive is going to be the same, because we have lowercase, 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 so that would be 2 out of 4, 50%. Okay? Again, in summary, anytime you see that you have heterozygous, that's always going to tell you that it is the dominant trait. Hope that helps with Punnett squares. Thanks for watching.
Okay, so let's do some of this together. Let's practice. So I want you to draw this box on your paper and divide it into 4 parts. I'll pause for you to do that. All right, let's match this up. So we have capital R, lowercase r. We'll just say this is dad, and then uppercase R, lowercase r on the top. We'll say this is mom. So let's match these up, and then we'll determine the genotype and the phenotype. So we'll have a RR, capital R, lowercase r, still capital R, lowercase r, and then lowercase, lowercase. So your box should look like this. Capital R, lowercase r. If you were doing it like the man in the video did, which he did it wrong, yes, you're bashing it up. So lowercase r, uppercase r. But anytime you have uppercase, you put it first. And then lowercase r, lowercase r. Remember, homozygous is the same, like 2 capitals or 2 lowercase. Heterozygous, it's different. So you have 1 uppercase, 1 lowercase. So I want you to think about these for a second, and I'm going to pause for you to do that, and let's see if you can get it correct before I show you the answers. So homozygous dominant, that would be 25%. Dominant is the capital. Recessive is the lowercase. So you have one of these tiles, which will be 25%, 1 out of 4, that is homozygous, the same, dominant, because they're capital. You have one that is homozygous, recessive. They're the same, and they're lowercase. And then you have 50%, 2 of these out of 4, that are heterozygous, half of them. So the dominant trait, you have 3. If it has an uppercase letter, it is dominant. So 1, 2, 3. 3 out of 4 would be 75%, and 1 out of 4, 25% would be the recessive trait. Let's do another one. Okay, so draw this out on your paper, and I want you to go ahead and fill in the Punnett square. Fill it in. I'll pause for you to do that. Okay, you should have uppercase T, uppercase T, uppercase T, lowercase t, uppercase T, uppercase T, uppercase T, lowercase t. Okay, so now I want you to think about what dominant and recessive meant. Homozygous, heterozygous. I'm going to pause for you to get some percentages here, and then we'll go over those answers. So your homozygous dominant, it must be the same, it must be capital. We have 2 of 4, so 50%. We have 50% that are heterozygous. We have none that are homozygous recessive because we don't have any that are both lowercase. So the phenotype here would be 100% dominant because if it has a capital letter, it is dominant. And we would have 0% recessive. All right, one more. So this time, I want you to not only fill in the Punnett square, I want you to go ahead and get these percentages. And I will pause and allow you time to do this. Okay, so you should have uppercase R, uppercase R, uppercase R, uppercase R, uppercase R, lowercase r, and uppercase R, lowercase r. So the homozygous dominant, the ones that are the same, capital letters, 2 of 4, so 50%. Heterozygous, 2 of 4, 50%. We don't have any that are 2 of the lowercase, so it's 0% homozygous recessive. And all of them have a capital in it, so it's 100% dominant for the phenotype, 0% recessive. Let's do another one. So do both, do the Punnett square and do the percentages. And I will pause for you to do this. All right, on this one, we just have lowercase n's. So we don't have any that have a capital letter. So we don't have any that are homozygous dominant. We don't have any that are heterozygous because we don't have that mixture. They are all homozygous recessive because they're all lowercase. So 0% in the phenotype for the dominant trait. They're all 100% recessive. So in summary, why are Punnett squares used in the study of genetics? Well, they're used for predicting possible genetic outcomes for offspring. They help to calculate the probability that certain genes will be passed on or not passed on to offspring. And as always, let's finish this up with a video.
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spend quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter, or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase little h. A guinea pig that does have hair can be represented as a big H, or a big H, little h. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h, is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H, little h is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype? Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H. We don't really know. We could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Or a percentage. 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. Well, what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. Now remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, little h, they have hair. So three of them here, the one big H baby and the two big H, little h babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. And let's do our exit ticket. When the allele combination is a dominant and a recessive trait together, which trait will be seen? Type your answer in the Zoom chat box. Of course, it is going to be dominant, because anytime you have an uppercase letter, the dominant takes over. All right, this is the end of lesson 8.8 and also unit 8.